Welcome, everyone. I'm Xinhe Yu, the chair of RMIT Professorial Academy, the host of this event. First, I would like to acknowledge the people of the Kulin Nation on whose unceded lands we are meeting today. I respectively acknowledge their elders, past and present. So today we shall hear from distinguished Professor Magdalena Plebanski, a lecture on cancer, aging, and vaccines. This is part of the activities by the Academy to fulfill its obligation as an ambassador, advocate, and thought leader for RMIT. So before we start, let's just get through some housekeeping matters. So this is a team live event. You will not be able to directly ask any questions via microphone. Please post your questions in the conversation section during the lecture. At the end of the lecture, I will pick up those popular questions to ask the presenter on your behalf until our lecture time is up. So let's just start the lecture by introducing the speaker. Distinguished Professor Magdalena Plebanski develops a new immune-based therapy and vaccine to optimize vaccination for the elderly, as well as to develop and validate new diagnosis, prognostics, and the treatments for ovarian cancer. She published extensively, including papers in top journals such as Science and Nature and Lancet. So many of Magdalena's invention has also progressed to human clinical trials or commercialization, and she has led this translation process in diverse roles such as inventor, CSO, CEO, director in biomedical, biotechnology companies nationally and internationally. So without further ado, please join me to welcome Magdalena to deliver her lecture. So over to you, Magdalena.
Thank you very much. That was a beautiful introduction. I also want to add my acknowledgement to the Woiwurrung and Boiwurrung language groups of the Western Kulin Nations, on whose land I sit right now. I don't know if my slide is presenting. I will. Am I sharing my slide? Sorry, just a question. Am I sharing my slide?
Yes, there's a slide, but it may not be in the. Yeah, okay. It's okay now.
So, so you can see the beautiful Mary Creek where we are, which is which is belonging to the Western Kulin Nations, Woiwurrung and Boiwurrung language groups, and which we are privileged to be on today, and from where I'm giving my talk. So, as well as being the director of the Biomedical and Health Innovation, and some of you may know me because of that, I also lead a fantastic group of researchers that are interested in cancer, aging, and vaccines at the Cancer, Aging, and Vaccines Lab at the School of Health and Biomedical Sciences in the College of STEM. Now, you may think, cancer, aging, and vaccines, how do they relate? So let's start with aging, and hopefully by the end of this talk, we'll get a clearer idea of why we're interested in those three topics together. So firstly, we're all going to get old. And in fact, not just individually, but humanity is getting older. And although this is not even across different countries, it's estimated that by 2050, in some countries like Spain, Italy, Japan, Korea, nearly a third of the population will be over 60. And in the older age groups, because life expectancy is also increasing, there may be even more significant increases in the percentage. End of the day, this increase in longevity, this increase in life does come with an increase in diseases that are associated with older individuals. And it is estimated, the WHO estimates, that there will be a 50% increase in cancer globally from 2010 to 2030. So that is quite significant. And we may ask ourselves, Why cancer, and why is it linked to an aging population? Well, as we grow older, the errors and damage in our replication machinery, and particularly at the DNA level, just probabilistically increase. It's cumulative. So over time, you have more of a chance of making an error as you're transcribing your DNA. There can also be mutations, and reactive oxygen species cause damage. And we know many of the environmental factors that can cause damage. For example, components of tobacco smoke, and this has been known for a while now. The other thing is that it's not just getting the damage, it's your ability to be able to repair and control that damage, because it's the mutations that allow cells to keep growing, proliferating, and not be eliminated by the natural controls of the body that are going to eventually result in cancer. And the immune system is critical to enable this control, and the immune system changes with age. In fact, the relationship between immunity and cancer has been known since the 1800s. In fact, Virchow said cancer lesions appeared in inflamed tissue in 1863, and more recently, it was characterized that tumors were called the wounds that do not heal, as in they are wounds that have an infiltrate of inflammatory immune cells. So already very early on, there was a possible relationship and causation in terms of inflammation driving or helping drive and sustain cancer. That's the bad part of the immune system. The good part is that, and again, it was Burnett in Australia who first said that logically, if cancers are appearing because it's inevitable that they appear, because it's inevitable that there are errors and there is damage, there has to be a mechanism to eliminate or inactivate such potentially dangerous mutant cells. And he postulated that this mechanism would be immunological in character. So the immune system and cancer are deeply related from the beginning, both in terms of potentially promoting and sustaining cancer in terms of inflammation, but also in terms of being able to control and eliminate it. So before we jump on to more specific things, I just want to give you a very quick idea of the immune system, because some of you may not be immunologists, and obviously as an immunologist, I absolutely love the immune system, and I hope many of you will be inspired after this as well. So first of all, the immune system has two arms. The first arm is the innate arm, and you can imagine it when an invader comes into the body. They're the first responders. And we have some cells there that are literally like bombs. And I put here a picture of a neutrophil. A neutrophil is filled up with granules, and it also eats anything that comes in its path. So say a bacteria comes into the body, it will just eat it until it explodes from overeating and release all these toxic granules. And so there's a lot of collateral damage, but it will eliminate bacteria, and it will act very quickly. As well as these first responders that just go for it, we also have cells appearing very quickly in this innate response that are designed to try to profile the invader and to call for backup. So these cells, again, most often are characterized by being able to take up whatever it is, bacteria, virus, cancer cell. And after they eat it, they profile the pieces, the bits and pieces from it, by showing them on their surface. So these are cells like macrophages, as well as myeloid-derived suppressor cells, or most importantly, dendritic cells. Now, dendritic cells, what they'll be able to do is to activate the rest of the immune system, the adaptive immune system. And the adaptive immune system what it's there is to really target the response specifically to that specific invader, to eliminate it in the best way, deploy, eliminate, but then also record the characteristics of that invader to be able to attack it far more efficiently the next time. Now, the other function, which doesn't get talked about often, but it's really important, is that the immune system is also designed to get down from the state of alertness and calm things down. So let's say whether in the initial phase where neutrophils and other cells are going crazy, releasing lots of inflammatory cytokines, that's fine. Trying to really eliminate whatever it is that it's attacking us. In the next phase, we need to not just deal with the infection, but get back to homeostasis and get back to a calm state. So as well as having cells, and these are T cells, that are able to, say, for example, kill infected cells, CD8 T cells, we also have within the CD4 T cells, cells that help other components of the immune system to generate the adaptive response, but we also have cells that just calm things down, regulatory T cells or Tregs. So now, back to cancer and aging. So what has been observed, though, is that even though there is an association of infiltration, and particularly this innate inflammatory association with cancer appearing, when you look at different stages, it's actually an immune infiltration that is made of adaptive cells can be associated with better survival. So it's which immune cells infiltrate the cancer and the ratio, what they're doing, what they're able to do, and how they cross-react with each other will be critical to the outcome from cancer. So, for example, your CD8 cells will kill mutated cells, can kill the cancer cells, and your adaptive Tregs can turn off the T cells. Now, their job is to bring things back to homeostasis. But if the Tregs get activated too early, they're bringing the body back to homeostasis before it has dealt with the problem, in this case, cancer. So how does this play out as we get older? Well, we have age-related dysfunction in our adaptive cells. So we are generating less newly formed, naive T and B cells. So our ability to recognize new targets decreases. The T cells themselves are less effective. And we have increased numbers of these regulatory T cells, Tregs, whose job is to turn things off. In terms of the innate immune system, they are more myeloid, that is from the innate system, than lymphoid cells being generated from the bone marrow. So we're having more of these inflammatory cells. But they seem to be less active. And I said to you before, one of the key roles of some of the innate cells, like macrophages, dendritic cells, is to eat things and then show them to the rest of the immune system. But with age, some of these cells have decreased capacity to eat or phagocytose. And they have increased pro-inflammatory molecule production. And we've mentioned that inflammation has been known for a long time to be associated with cancer. And now we know that one of the reasons that it is that some of these pro-inflammatory molecules also are molecules that attract additional cells into the tumor environment. So they're pro-inflammatory chemokines. And these pro-inflammatory chemokines may be attracting immune cells to the environment, but they're the wrong kind of cells. In my picture here, we have a pirate. So that's not whom we want to attract to the tumor environment. We want to attract the cells that are actually going to deal with the tumor rather than just have a monkey on their shoulder. So, immunity, aging. So what can we do? Well, we know that there's a skew in the ratio. So there's more Tregs and less CD8s in old age. There's also less capability to recognize new targets. So our natural immune system is not really coping very well. So somehow, how do we increase the CD8 to Treg ratio? Well, just to exemplify why this is so important, if we have more CD8s, they kill the tumor. If the Tregs come in, they turn off the generation of more CD8s. They turn off also the function of the CD8s. And then the tumor itself will be secreting inflammatory chemokines, which don't only promote tumor cell growth, including tumor stem cells, but also attract further Tregs into that environment. So it really compounds. What's happening naturally is just not good enough. So we need to increase that CD8 numbers and have them target the tumor. So one way of increasing CD8 numbers will be to generate CD8s with a vaccine and cancer-specific CD8 T cells. Now, unfortunately, as far as cancer vaccines goes, traditionally they're inactivated or attenuated whole pathogens, or have been in the past, and we'll come back down to that in a minute. And also, most of the subunit vaccines that don't use a whole pathogen, say a whole bacteria, a whole virus, the subunits have added an adjuvant. This is something that will just irritate the immune system, will make it wake up. But usually, it's a pro-inflammatory adjuvant, and we just said that inflammation is bad. So what would we want to do in cancer, and for this in older individuals, is to increase CD8 T cells that recognize tumor antigens, decrease the Tregs somehow in the tumor environment, or decrease their activity in the tumor environment, and try to do the above without increasing inflammation. So, could nanotechnology offer a solution? And we think so, and we very much think that it can. And this is just an example from some of our work, but this is now has expanded to others studying this area very intensely. And a long time ago, we actually found that defining the size, shape, and charge of nanoparticles made them deliver the targets, the antigen to these dendritic cells, and without adding any adjuvants, any pro-inflammatory factors, by delivering the antigen, the target we wanted to attack, to the right cell in the body, right immune cell, we could elicit massive immune responses. And these responses were able in preventive models to prevent cancer challenge when 2 months after a single immunization, as well as rapidly clear, that was within 2 weeks, an already established cancer. So this has resulted from our many papers. Over 100 will be in this particular area of vaccination and nanotechnology, and multiple patents. But the point is that it is possible, and it was already seen to be possible some time ago. This particular couple of papers have been cited over 1,300 times. So it is possible, and now many people are constructing this to induce immunity, and to do so without conventional inflammation, because we want to avoid that, particularly in settings like cancer. And talking about vaccination, if there was a time to talk about vaccination and to open this as an opportunity for cancer, the time is now. Just to give you a quick historical perspective, it was in the 1600s that we have a record of the first big campaign coming from China's Emperor Kang, endorsing the inhalation of ground-up smallpox scabs for protection against smallpox. Now, following the generation of many vaccines and other approaches across the world, smallpox was eliminated around 300, almost 400 years later. So it's taken a while. But then if we think about measles, for which the campaign started much later, we actually were able, as humanity, largely eliminate measles from the world, particularly in the USA and the Americas by the 2000s. Polio, which did include a massive eradication campaign, has been eliminated from the Americas and Europe in 2002. And whether all of this progress has been very slow and mainly focused, as you can see on the list here, in live attenuated vaccines and irritant adjuvants like aluminium salts, you can see that other things have started appearing. So in the 2000s, we have adjuvants MF59 and AO03 approved for some influenza vaccines, and I'll come back to those in a minute, as well as virus-like particles. Now, again, these are nanoparticles, but they're made of pure protein, and that's the basis for the papillomavirus vaccine Gardasil, which is preventive for cervical cancer. Also, more recently, very recently, what has been approved is basically feeding your dendritic cells outside of the body, your own dendritic cells, with bits of the prostate cancer, and then putting them back in your body to stimulate the immune system. And this kind of dendritic cell-based therapies are a very active field of research right now. And in India, as recently as 2017, that has been approved for the treatment of multiple cancers, this personalized dendritic cell-based therapies. So there are other ways of inducing immunity other than attenuated or inactivated pathogens or combination with aluminium salts. But progress has been slow until COVID. And COVID-19 has galvanized the approaches that have gone and been progressed to human clinical trials in parallel, with hundreds of vaccines and new vaccine approaches now having been tested in humans. The ones that have really progressed further, which I have on the right-hand side and which have been licensed for emergency use across different countries, I would note that they're pretty much the ones that made it, except some of the ones which have still been based, some of the ones that have come out of China, which have still been based on inactivated pathogen, the whole virus itself. The other ones really have used new technologies. And in fact, 3 of the vaccines that are now approved in Australia are all nanoparticles. They're all in the what I would call correct size range to be interesting to dendritic cells to eat them and show them to the immune system. And they're triggering, some of them, novel danger signals, which are not as pro-inflammatory as conventional danger signals. So there is, for example, a molecule that senses danger inside cells called TLR9, and that will sense the RNA in it and help stimulate the immune response. But it is an alternative type of stimulation to really providing a large inflammatory danger signal. And I did promise you to mention Novavax, and I would say that that's a very interesting vaccine that's going to come at some point, which uses a new adjuvant. But this new adjuvant, again, is not a conventional inflammatory adjuvant. And it, again, is a particle, and it tends to promote what's called lipid body cross-presentation, which allows it to stimulate CD8 T cells effectively without engaging conventional adjuvant pathways. And that particular alternate way of stimulating the immune response also has been seen to be very effective in older individuals. So we now have vaccines, and obviously we also have the adenovirus-based vaccines that we are seeing having efficacy in older individuals, which opens real promise for the development of cancer vaccines. And within these, we're also seeing some vaccine approaches that are avoiding or not needing to engage in a strong way the conventional inflammatory pathways. And one of the things I would like to also talk about, if we are going to be going and developing new vaccines, we have to really look beyond just what the vaccine does to protect against disease that you wanted to protect against. And there might be opportunities to train your immune system in other ways. And in cancer, what is very interesting is that this has been happening for a long time now. The BCG vaccine, which is a vaccine to treat tuberculosis, to prevent tuberculosis, is used as a treatment for bladder cancer. So one vaccine is having a different use. And how does it do it? It's still being debated. But one could think that it is engaging some of these innate immune mechanisms that are able to really be somehow specific for the cancer, and an active field of research. So there are other opportunities, not just in terms of specific vaccine approaches, but also non-specific vaccine approaches. And linking back to what we were talking about, cancer, aging, and vaccines, we really need to acknowledge that vaccines, and both in terms of specific immunity that they elicit, but also the non-specific immunity, will be different in different individuals. And on the left, we're just showing some of our recent papers, but this is a very active field of research. And across all of these papers, what we are seeing is that it's not only age that is important in terms of vaccines, it's also sex and gender. And females, particularly older females, have higher inflammatory responses. So women are set up to have a more pro-inflammatory profile in the immune system generally, and this can be linked to sex hormones or X-linked genes, as well as other factors. Even the gut microbiota can be different across sex and gender, and the microbiota, the microbiome, will also influence your immune system. So we are having an indirect but strong effect on the immune system as well. It's also not quite understood why, but it has been observed that chronic infections with viruses and parasites are more prevalent in males, and it has been posited that this will alter vaccine efficacy. So what we need to do is to really understand both of the specific and non-specific effects. Now, I'm just giving you one slide of some of the current work at RMIT. So we're running a large-scale human trial to understand vaccination against the two vaccines used the most in the elderly, influenza and DTP vaccines, and understanding how they induced immune responses with big data. So looking at this whole spectrum, this whole training of the body by the vaccine in terms of being able to deal with infections with big data analysis. So trying to understand not just the specific response, those influenza-induced and antibody against influenza, which is where many other trials stop, but looking at whole body effects and effects on the all of the genes that are involved in the immune system with RNA-seq, epigenetics, and profiling, also looking at the microbiome. We're similarly running vaccine trials of COVID-19 vaccines in vulnerable populations to determine the effect of age, sex, and pregnancy on immune homeostasis and activation by COVID-19 vaccines, again using big data analysis, as well as a study of long-term effects of COVID-19. This is not a vaccine study, but it's more to understand the effects of COVID-19 infection itself on immune homeostasis, inflammation, and autoimmunity. Because us, as well as others, we know where we are now in research and in terms of what we hope to be able to offer to the next generations, which is personalized vaccinology, where the impact of your age, your sex, your gender, your background generally of your immune system is considered when recommending or applying or developing vaccines, so they are truly effective. So, back to cancer. Let's assume now we can make a vaccine. So what should we target? And again, these are just examples from our own research, but many other people are working in these areas. And I've divided the targets in cancer into 4. One is targets that come from immunoprivileged sites, which is either because they were expressed while we were embryos and then they stopped being expressed, some proteins, and then they're re-expressed in cancer. So your immune system hasn't seen them. So if they're shown to the immune system, it's going to be more able to attack it because it won't think that it's your own body, it's your own self, and therefore they're good targets to attack. So some of these molecules that are expressed throughout ontogeny may be associated with cancers that are able to in turn suppress the immune system. So they would be very good targets to get rid of. We also have autoantigens because cancer cells are really our own cells. And there are some ways of ramping up some antibodies that can recognize these self-proteins, which may not be expressed in high quantities in normal cells, but they're expressed in high quantities in cancer cells, which gives them their specificity. We can also modify the actual targets. We can change the sequences of the proteins that we put into the vaccines so that we make them more potent and we make the immune system more interested in generating an immune response against them. There's also, in the advent of personalized medicine, there's personalized neoantigens, which means as your tumor changes and mutates, it generates new targets. So if we can sequence those targets, we can have personalized targets for an individual tumor. And that should expand our CD8s and hopefully change that negative Treg to CD8 T cell ratio that we normally have. So let's assume we have them. You know, there's a way forwards for the field to have a potent vaccine that works in older individuals and to identify the targets that we want to put in them. There is still a problem we can generate these CD8 cells, but they might just stay in the periphery and not migrate into the tumor. And why is that? Well, the chemokines, the inflammatory factors that are produced by the cancer cells and the initially infiltrating cells tend to attract Tregs. So the cancer cell is attracting Tregs to its environment. And why is it not attracting CD8 cells? Well, even if some immune cells are in there and trying to attract CD8 cells, the cancer itself is secreting molecules, proteases, that chop up the molecule that can attract the beneficial CD8s. So some approaches that we are doing at the moment are to try to repurpose drugs that block that activity of the cancer cell. So playing mind games and trying to get ahead of the tumor. So the tumor is destroying the molecule that prevents CD8s from migrating into the tumor. Okay, let's prevent the activity of the molecule that blocks that migration. We also have found new pathways by which to attract CD8 cells into the tumor. And with that, we should be able to change the ratio of, so generate more CD8s in the environment, attract them to the tumor, and then what happens? Unfortunately, there's already many Tregs in there. And not just that, the tumor cells as well as the Tregs express molecules that turn off the beneficial activity of the CD8s. And in fact, the Nobel Prize in 2018 was given for this discovery, the discovery of checkpoints and the possibility of targeting this checkpoint molecules with inhibitors as a cancer therapy. So here you can see that, say we have your CD8 cell being blocked from killing the cancer cell because it's interacting with this molecule called PD-L1. And monoclonal antibodies against either PD-L1 or PD-1 therefore deblock the activity of the CD8, so it can kill the cancer cells. That will stop inflammation from happening, which will also help the CD8 cells to keep being in that environment and not have more Tregs migrating in there. So it's a whole dynamic structure. Now we're involved in a trial right now testing one of our prognostic biomarkers, and also at the end of the trial, together with our collaborators from WEHI, AstraZeneca, and Anscog, we are using checkpoint inhibitors. So we have the whole combination stream to try to offer benefit to patients. And offering benefits to patients is a passion for us. And our focus is on ovarian cancer. And why are we so passionate about ovarian cancer? It's because it's got really high mortality. And it's not just about having better cancer treatments. The problem is that it's detected too late. And it has vague symptoms. And even if you pay attention to your vague symptoms, there are no biomarkers at the moment that you can go to the GP and say, yeah, you've got early stage ovarian cancer. With early detection, survival is expected to be 90%, over 90%. But most women are detected late, where survival is between 5 to 10%. And after initial treatment, most women, over 80%, get a lethal recurrence. And this is what's called platinum-resistant disease. And there are few other options for treatment. So there are big needs in ovarian cancer, not just to develop new treatments, but also to develop new diagnostics. And we believe that whether the previous time period in cancer research has focused on personalizing treatments and diagnostics even to the genetics of the cancer, as well as changes in genetics of the cancer. The next stage will be about individualizing treatment, also thinking about the immune system. And we believe, and we run projects in the lab across all stages of early detection, personalized therapy, and innovative new treatments, focusing on including the immune system across all these stages. And this is already yielding some really exciting results. And our dream, and enabled by collaborations, so we have a collaboration with Professor Anand Mitchell on practical sensors, which we hope down the line at some point may enable diagnosis in the clinic or even at home from a single drop of blood. And we find the biomarkers for early detection, and we have some really excellent candidates, and they develop the technologies which hopefully will make it very practical to really go that step of early detection. And these same sensors may be used for personalized therapy, personalizing therapy. And again, if there are biomarkers that can tell us which drug will be better to use in which individuals, then from a drop of blood, we can recommend the best treatment that will have the highest possibility of acting on that particular tumor in that particular person that has a particular type of immune system. We're also collaborating with Professor Suresh Bhargava for innovative new treatments in terms of new drugs that are able to target this platinum-resistant disease. So even if everything fails in the initial stages, there is still a drug at the end that can be used and treat this platinum-resistant disease. We could avoid all this, though, if we were able to activate the immune system beforehand to the targets that will then be able to protect from the very beginning against the natural generation of the cancer in the first place, by having our immune system do the job even before the cancer starts. And that is really the dream for us. Lastly, when talking about personalizing and when talking about the immune system, we really have to consider that the immune system, and even if it helps us in diagnosis, here I have a picture of a black hole. It's an artist's impression of a black hole, modifying the space around it. And that's how we can see a black hole. So the immune system can be considered the space around the cancer, and we can see the changes in the immune system, and that can help us with the detection. It can also help us in terms of treating the tumors. Having said that, the immune system is altered by a wide variety of factors, including mental health, and like we mentioned before, microbiome, so behavior, diet. If we really, truly want to understand the immune system, we have to be intrinsically multidisciplinary and understand our patients and our immune system as a whole, as an interacting system within an individual. So for the future, and the way I see the future emerging for cancer generally, not just for ovarian cancer, is that there will be more of a focus on immunotherapeutic drugs. We are being told we are living the immunotherapy revolution in cancer, and that is truly what we are seeing around us. We will also see a lot of repurposing of drugs, combining of drugs, personalizing treatment to genetic subtypes, but also to immune status, and that has me very positively excited that it will have a real impact and enable earlier diagnosis, and perhaps in the future, that may not be too far away, promote vaccine development, which will enable the new generations not to have to worry about getting cancers such as ovarian cancer. And for that, we really need to focus. So the way I see cancer in the future is very much about personalized detection and treatment and a real focus on multidisciplinarity. And just to finish up, so we have plenty of time for questions, I wanted to mention one of the reasons I was so excited to come to RMIT has been my other job, which has been the Enabling Capability Platform Director, because that other job is about bringing together researchers across multiple disciplines to work on projects that engage externally for impact. And that is a tremendous opportunity for us to really understand how together across disciplines we can solve major problems such as cancer and aging, and to really galvanize vaccines to be able to do so. So just as my last slide, I want to mention some of my collaborators and our amazing lab at RMIT, who makes all of the research possible, but also keeps all of us motivated and excited to make a difference. So thank you.
Thank you very much, Magdalena, for that fascinating talk. So we have a few, so one question come along, but I think there will be more. I might just, so this is a very general question, I mean, certainly give like people like me, engineer, have no sort of too much knowledge about, vaccination, cancer cells, etc. So this is certainly sort of eye-opening. So let's try to be not sort of too political, but just in the way of going, you think that the way we are dealing with, the current COVID is okay or too rigid, or the way we proceed, the strategy we have?
I've actually been very impressed by how quickly vaccines were developed. And as I said initially, initially we were talking about hundreds of years, then decades, and this is just to develop one vaccine and using approaches that are really not inducing the best type of immune response. So, for example, alum, which is an adjuvant put in many vaccines, the responses that it induces are okay and they reach the threshold for protection, but there are many other better adjuvants out there. But there has been no willingness to really say, okay, what is the best adjuvant? Is there a way in which we can make things better? There has been a lot of inaction. And while COVID-19 is absolutely terrible, what it has done is it has moved the field forwards in terms of being able to look at vaccines in a comprehensive way, really put their safety under the microscope, and accelerate development by an injection of funds. So I think the opposite. I actually think it has been one way in which humanity and multiple countries have shown that they can respond to a challenge that concerns all of us globally. So I'm positive rather than negative about the fact that vaccines were developed so quickly.
Okay, thanks. We've got quite a few questions pouring in. So here's the one. I have always wondered how much nutrition play a role in the prevention of cancer as well as recovery from this type of cancer. What role do you see nutrition playing in regard to cancer?
That is a beautiful question. Thank you so much for that. It's very important, both directly and indirectly. So if you remember, we were talking about inflammation bad when it comes to cancer. And there are gut bacteria that basically promote factors that decrease inflammation in a healthy way. And having these anti-inflammatory bacteria in your gut is very helpful. We actually published a paper in that is the immunity paper that we just mentioned briefly, showing that the gut microbiota and the composition of the microbiota actually influences response to a cancer drug called cyclophosphamide, which gets used in lung cancer and which gets used in ovarian cancer. So the composition of your gut microbiota is directly influencing the patient's response to chemotherapy. So it's absolutely plays a role, and it's important. And thank you so much for that question. Sorry. And your gut microbiota, just as an aside, so the composition of your gut microbiota in turn is influenced by the diet. So particularly fibrous diet tends to promote the bacteria that can have these positive effects.
Okay, so here's another one. So you mentioned the tailored therapies, and I'm wondering if you think vaccine will need to be tailored too.
Yes, that's a big yes. Personalized vaccination. Having said that, it depends how much you personalize. So there'll still be groupings, but I would say we're seeing it even now. We have different types of vaccine recommendations that were had for older and younger individuals. And that's fair because the immune system of older and younger individuals is different. I would see in the future we can go a little bit more granular, but it still won't be, your own personalized formulation for just you, but that we can be more nuanced and have a real optimized safety and immunogenicity profile, for example, for pregnant women, or for example, for the immunocompromised. And those things are already happening. So we will have more and more personalization of vaccines to particular groups and hopefully to particular vulnerable groups as well.
Okay, here's another one. This one appraised your very informative talk. Let's go to the actual question. So you mentioned the DPP4, and this could be a potential drug target to help and regulate CD4 cells. You also mentioned the checkpoint inhibitors. I'm wondering what inhibitors of DPP4 or CTLA4 or PD1 or PD1-L1 exist and has been trialed for CD4 upregulation. And you mentioned antibodies as checkpoint inhibitors.
Okay, so a very specific and long question. So in terms of your first part, the blocking metalloproteinases activity, it's an emerging field. And at the moment, there is a drug called sitagliptin, which gets used to treat diabetes, and that is something that we and others are looking to repurpose to inhibit DPP4. So there has not been a large-scale human trial on that yet, but it's a safe drug already used for a different indication. So that's where there is a push towards repurposing drugs because that can give us an outcome faster. For the second part of your question, a lot of the different big pharmas all have their own checkpoint inhibitors. And we have, of course, anti-PD1, anti-PD-L1, anti-CTLA4, some other ones like anti-LAG3, and different companies are progressing them separately. Now, there have been studies of these checkpoint inhibitors alone or in combination with each other, as well as alone or in combination with other therapies. And what has been emerging is that while the checkpoints can be absolutely amazing for some cancers by themselves, for other cancers, what's happening is that they are amazing, as in they can give cures where previously there were no cures in cancer. They need to be combined with other drugs, sometimes existing drugs, sometimes novel drugs, to really provide maximum benefit to the patients.
Okay, thanks. Here's another one. Quite a few is coming in. So did focus, money, and resources put into development of COVID vaccines result in the boost for research and development of other vaccines?
Indirectly, 100%. Indirectly. In terms of directly, no. If anything, there's more competition. And but indirectly, and what I see into the future, just having such amazing insights into the mechanism by which different types of old and new vaccines work, safety profiles, how they work in different populations, is going to give us information we're going to be working on for a very long time. So indirectly, yes. In terms of directly, that's going to be for us to exploit as we go forwards, and hopefully it will be exploited in fields like cancer.
Okay, here's another question from engineer computer science. He said, you mentioned you are taking a big data approach. What methods have your team used, and what barrier have you encountered with those methods?
Yes, great question. So in terms of big data, we do RNA-seq, we do microbiome, we do epigenetics, we do a cellular immunology analysis like flow cytometry. We've got big data profiling for cytokines and chemokines, using things like bioplexes. We're also setting up to do single cell RNA-seq. So a few approaches there. We're looking forward to collaborating to do metabolomics as well. So anybody out here who's interested. What barriers? Well, we've had to set up a lot of things at RMIT. But now they're running. So we set up an epigenetics laboratory. So it looks at the methylation and changes in the DNA and tells you which genes have a propensity to be expressed or not. We set up a biobanking for human clinical trials as well. So that's running. So a lot of the things in terms of barriers, I would say it was just about galvanizing and building. But now it's here, and I wouldn't say a barrier. Everybody has been very helpful in terms of being able to set up these things, but it's just taken some time and effort to get things running.
We're just about time, but let's just the last two questions. There's a small, quick one, right? Is there opportunity to join the trial you mentioned earlier?
Okay. Email me separately about that. Yes, so there are open trials. There's still recruitment happening in the trials I mentioned.
Yeah.
Having said that, each trial has its own set of parameters, and it's the clinicians that will assess eligibility criteria. So email me separately.
Yeah, so this is the last one. There's quite a few others. I'm sorry, I can't go through all of them. So there have been recent developments in cancer therapies where small molecule drugs are tethered to protein degrader. Could or has a similar approach being taken with vaccines?
Oh, sorry, I just didn't catch the middle bit. So similar approaches with small molecules that do what?
Are tethered to protein degrader, PROTACS.
Okay, so I think the question is about targeting approaches to the tumor. Yeah. And absolutely, and there are many ways of doing that. Not just antibodies. There are other ways of targeting molecules to the tumor itself, which are related to how things drain in the body. So the vasculature of tumors is different. So some things tend to accumulate in tumors because of that. You also can target specific molecules on the surface of cancer cells, of course, and get your accumulation of substances like that. You can also use combined imaging and treatment approaches, what's called theranostics. So again, you're imaging and targeting at the same time. Now, once you have tumor destruction in a particular way called necrosis, that in turn can trigger more of the immune system being activated. So even standard chemotherapy at some point will involve a component of immunotherapy, even if it isn't directly designed to be immunotherapy. Hopefully I answered that question.
Thank you. I think our time is up. There's still quite a few questions we haven't responded. So what we do is we will collect those questions, and then I think in the transcripting, I think we will ask Magdalena to prepare a response. So for that, thank you very much, Magdalena, for a fascinating talk, and also thank everybody else to attend the seminar. So thank you very much.
Thank you for the opportunity.
Okay.
And thank you everybody for coming.
Thank you.